So my second talk will be on exotoxin. In the first talk, we talked about the endotoxin as a part of the virulence factors which are responsible for the disease causing, and here we talk about the exotoxin. Now, exotoxins are more severe. They are the protein toxins of bacteria, right? And in exotoxins, like proteins, mostly enzymes secreted by the bacteria. Unlike lipopolysaccharides, are lipid molecules in case of endotoxin. So in exotoxin, the major part is protein, or it is part of a protein released from the bacteria, and these bacterial proteins are mostly enzymes, enzymatic activity secreted by the bacteria, which may cause the damage to the host cell, a different part of the host cells. It produced during the growth and metabolism of the bacteria. It secrete into the medium or release after the lysis. So it may also secrete, or it may also produce after lysis of a bacteria. For example, Salmonella, it can lyse and produce the bacteria toxins. Now found in gram positive and gram negative bacteria both, but more common in the gram positive bacteria. Unlike the endotoxin, where it was most common, are found in the gram negative bacteria. Now, genes for exotoxins often carried on plasmids are bacteriophages, right? So, genes for exotoxins often carried on plasmids and bacteriophages, and highly toxic and specific, act on particular cell type or tissues. Diseases are due to the toxin action, not the bacteria itself. So, it is not the bacteria which is causing the infection, but the bacteria release toxins, right? Release these protein and enzymes which damage to the host machinery or host system, host cellular system, to damage the host. For example, botulinum toxin, Staphylococcus food poisoning bacteria, right? So the disease is caused by these toxins. So the protein, here is a protein. Enzymatic natures act as a repeatedly very small amount of lethal dose. For example, in several microliter of molecules, 0.001 microliter. So this is a very small amount of the toxins are required to. So we have to check this through LD50 or LD90 or LD100. Lethal dose 50. It means what is the, for example, if LD50 LD50. LD50 is microgram. Then we have LD50. Suppose this is a toxin 1, toxin 2. So which toxin is more lethal, more severe? This toxin is more severe because it, in the minimum quantity, less quantity, it will reduce or it will kill the 50% of the laboratory animal. Whereas this is 10 to the minus 2, little higher. So we just check through this LD50, lethal dose 50 calculation. You can check the toxicity or you can check the severity of the toxins. The soluble in body fluids. So it is important that it need to be solubilized in the body fluid. It diffused and is spread quickly so that it can cause the toxic effect. Among the most lethal substances, right, known as 1 milligram botulinum toxins, 1 milligram toxins can kill 1 million guinea pigs. So this is just like estimation. In a very small quantity of the botulinum toxin, Clostridium botulinum bacteria which release this toxin, it can kill the millions of guinea pig as a laboratory animals. Exotoxin effect are disease specific, right? For example, in case of tetanus toxin, it causes disease, right? It causes disease and leading to the death of the person. Immunity toxin, immunity and toxoids. So how do you maintain the immunity against these toxin? How these toxins can be neutralized, right? Just like vaccines are used to neutralize the antibodies of a particular antibodies are used to neutralize the various pathogen. Antigen surface of these pathogens neutralized by the these antibodies raised through vaccine or through the microorganisms directly. So immunity can be developed, can be maintained on this, and whereas the immunity cannot be maintained against the exotoxins because they are lipid membrane. So body produces the antitoxins like neutralizing antibodies against these, so immunity can be produced. Toxoids can be generated, can be developed. Toxoids means like exotoxins inactivated by heat, formaldehyde, iodine, etc., and non-toxic but immunogenic, used in the vaccine. Example, diphtheria, tetanus toxin, diphtheria, tetanus, toxoids, DPAP, DTAP vaccines, right? We call diphtheria, tetanus toxin vaccine. So these vaccines can be developed against these toxins. Toxoids can be produced by attenuation process. So all these things are possible in these things. Now, what classification of these exotoxins we call AB toxins. AB toxins are consist of a two polypeptide subunit, A that is active and B for binding. Now, enzymatic toxic effect caused by the A, because A subunit is something which cleaves from the toxins and it binds to the surface where the toxin can get enter and show the enzymatic activity, because whole toxin may not have the enzymatic activity, but one of the part of this toxin, one part is binding part, another part is enzymatic action part, which cleave the membrane, which cross the barriers and get enter and find to the target for block the protein synthesis, right, or block the nerves impulses and so on. B is the binding, right? It enter into the cells and disrupt the host metabolism. Now, for example, diphtheria toxin, Corynebacterium diphtheria. So diphtheria toxin is a example of toxin which is very severe toxin, and it get enter into the cell and it cause the disease called diphtheria, right, caused by Corynebacterium diphtheria. And generally it is a child disease where the person cannot muscles can block and you cannot eat, you cannot properly breathe and so on. Whereas the cholera toxin is Vibrio cholera is one of the bacteria, gram negative bacteria, which cause the cholera, and this cholera toxin is produced leading to the diarrhea, loss of water, and then acidosis, then anaphylactic shocks may take place, blood acidity may pH get reduces and so on. So all this caused by these toxins. Botulinum toxins are, tetanus toxins are Clostridium species, Clostridium botulinum, Clostridium tetani. They are the toxins which are called the neurotoxins, and cholera toxins are the enterotoxins because it acts on the intestine, and botulinum toxins it act on the neurotoxins, and it kind of control the muscles spasm. It basically lose the control of the muscles regulation, and the muscles regulation is disturbed, and the spasm take place, and the diaphragms get ruptured, and the whole respiratory system will be blocked and will be broken, and the patient's death may take place through that. It's a very severe toxin, both botulinum toxin and tetanus toxin. The bacteria is not going to harm Clostridium botulinum, but the Clostridium botulinum, when they release a toxin, release toxin itself, they cause damage to the host cell. Now then we have genotoxins. Genotoxins are cause the DNA break, mutation, cancer. Example, cytolethal distanding toxins, typhoid toxins, and then colibactin toxins. So all these genotoxins cause the damage to the cell and damage to the DNA in the cell and genome through mutations and through cancer. The membrane disrupting toxins, there are some category of the membrane disrupting toxin. In membrane disrupting toxin, we have causes the lysis of the host cell by damaging the plasma membrane. So because plasma membrane is a barrier, so if you lyse the host cells by damaging the plasma membrane, then the host cells are more prone to get these toxins, or these bacteria which release the toxins, and they may damage the whole host system. Right. The mechanism is simple: transit pores. These pore channels are formed in this membrane like this, and this pore formation takes place, and we call the protein channels. Through these protein channels like this, so this is the channels. For example, this is the channel. So this is how it can get entered through these channels. and then phospholipid disruptions takes place. So phospholipid disruptions means like you can disturb the phospholipid and get enter. So basically it is the mechanism how to break the barriers and get find the routes path to get enter into the cell and cause the damage. For example, leukocidin, it killed the phagocytes, right? All the phagocytes kill and then weaken the immunity, because these phagocytes helps the bacteria to phagocytose, the virus to phagocytose, and after phagocytosis they can engulf these cells. But once you kill the phagocytosis process through leukocidin, then in the leukocyte, then the immunity is getting weaker, and it produced by the staphylococci, streptococci. So all these cocci chain forming bacteria, gram positive bacteria, they can produce these toxins and kill the cells. Hemolysin is to destroy the RBCs and release the iron, and then streptolysin O is oxygen labile, and streptolysin S is oxygen stable. So in the oxygen, in the absence of oxygen, it works streptococcus or streptolysin O, whereas streptolysin S is stable, and so it is in the presence of oxygen it works. Now the Clostridium perfringens alpha toxin is basically, Clostridium perfringens is something which causes the gas gangrene, and this toxin destroys the phospholipids of the membrane, and its role is help in pathogen escape phagocytosis. So it also allows the act as anti-phagocytosis. It does not allow the cells to involve in phagocytosis of these cells, and these bacteria escape from the being phagocytosed. It is spread through the tissues. So, and once it is not phagocytosed, it is not phagocytosis is blocked, then it can spread through tissue or cell. It also produces some intracellular pathogens like Listeria, Trypanosoma cruzi to escape from the phagosome. So they also escape from the phagosome through these means. Then we have a super antigens, and super antigens act by the overestimating the immune system. So you can also overstimulate the immune system. We call immunomodulation. So this is another way of treating patient or infection control is the immunomodulation. So there are several immunomodulator has come into the market, and these immunomodulator can be used to develop the immunity or to increase the immunity. Our immunomodulation process is also being carried out through the therapeutic vaccines also. We can learn the immune system how to behave, how to act against such kind of a bacteria. This is again done by immune modulator. So act by the overstimulating the immune system, and the mechanism is bind directly to MHC2 T cell receptors. It causes massive T cell activation. So when the T cell get activated, it causes the cytotoxin storm, IL-1, TN-alpha, IN-alpha, gamma, etc. So this mechanism is basically produced, and we escape this, and this immunity get weaken and weaken. It causes the fever, it causes the nausea, vomiting, diarrhea, shock, and even death. It depends on how severe the infection is. Then we have example of these superantigens are staphylococcal enterotoxin, that is food poisoning, then toxic shock syndrome toxin like TSST1. So the name of the toxins are by target cells, like depending upon the site of action, we call them as we name them these toxins like neurotoxins. Neurotoxin acts on the nerve cells. Cardiotoxins on the heart tissues and heart cells, valves. Hepatotoxins are the toxins which act on the livers, and leukotoxins which acts on WBC, white blood cells, and enterotoxins on GI tract, gastrointestinal tract, and cytotoxins, they are the general toxins which can act on any cell. So by disease caused diphtheria toxins, tetanus toxin are the diseases. And by producing the bacterium, the Clostridium botulinum called botulinum toxins, and Vibrio cholera release the Vibrio enterotoxin. So these are some name given on the basis of the Vibrio or bacteria through which the toxins are produced, or the site to which the toxins acts on. So this is the example of exotoxins. Exotoxins are toxic substances released outside the bacteria, like here, they are releasing out, and so this is Clostridium botulinum example, and protein produced by the pathogenic bacteria, and they cause cellular toxicity. Representative exotoxin diseases, the Clostridium botulinum toxins like botulism, neurotoxins, flaccid paralysis. So this Clostridium botulinum may also cause the this kind of a symptom like a flaccid paralysis because it is a neurotoxin, so it causes some damage, right? Clostridium tetani is a tetanus toxins, tetanus neurotoxins. This is spastic paralysis takes place and the muscles get a spasm and ruptures, especially the diaphragm and the lungs muscles. So that basically doesn't allow the respiratory system to work, and whole respiratory system get collapsed in this case, and the person can die. Corynebacterium diphtheria is a diphtheria toxins. This is a diphtheria toxins, is cytotoxins inhibit the protein synthesis, and that block the whole process of protein synthesis, and person may die. Vibrio cholera is cholera, which is enterotoxin because it enter into the colon or enter into the intestines, so we call enterotoxin. Generally, it is watery diarrhea caused by the Vibrio cholera. Streptococcus aureus, enterotoxin, TSS1, then TST1, then food poisoning, toxic shock syndrome, and so on. So these are some representative examples of the exotoxins and diseases. How exotoxins causing the disease, or how disease get established through these exotoxins. So in these exotoxins, it is not the bacteria which is caused. Bacteria can release either, or bacteria can release not, but bacteria can be present in the system and cause the release of these toxins. Now, these are some examples of disease caused by the exotoxins. For example, botulism or tetanus. So botulism is caused by the Clostridium botulinum. This is called type of exotoxin A and B. And the mechanism of action of these botulinum toxin is a neurotoxin. It prevents transmission of a nerve impulse. Flaccid paralysis takes place because of this. In tetanus toxin, the Clostridium tetani, again AB type exotoxins, it causes the neurotoxin. It blocks the nerve impulses to the muscles relaxation pathways and results in the uncontrolled muscles contractions, and that kind of diaphragm muscles get ruptures and person may die. Corynebacterium diphtheria is a bacteria which causes the diphtheria, and the disease called diphtheria, and the symptoms are a mechanism is cytotoxin inhibit the protein synthesis, and especially in the nerve, hearts, and kidney cells. So completely block the functioning of these nerve and heart muscles and kidney. That cause organ failure takes place and person can die. The scaled skin syndrome caused by Staphylococcus aureus, and again AB type. This is basically exotoxin causes skin layer to separate and slough off. The cholera toxin is Vibrio cholera, is AB type of toxins, are enterotoxins, causes the secretion of a large amount of the fluid and electrolytes that results in the diarrhea. Then we have traveler's diarrhea. The enterotoxigenic Escherichia coli and Shigella species are responsible for the traveler diarrhea. This is again enterotoxin caused by secretion of a large amount of fluids and electrolytes that results in the diarrhea. So all these disease-oriented infections are disease-causing pathogens which may cause disease through the release of these toxins, through the release of these protein or enzymes. The anthrax is basically Bacillus anthracis. Two A components enter into the cell via the same B. B is like binding proteins, and A protein causes the shock and reduces the immune responses. By reducing the immune responses, it activates the bacteria to further grow in the cell. Gastric stomach cancers may be caused by Helicobacter pylori or Helicobacter species. This is the genotoxic causes breaks in the DNA, and genotoxic causes break in the DNA. Then skin and soft tissue infections may take place. Methicillin resistance SRAs, membrane disrupting, and the penton valentine leukocidin found in the community acquired strains of MRSA, which makes the pores in WBC membrane and cause the kind of a porosity and the lysing of the cells, and that will reduce the immune system, and the immune system and reduce the infection prone. So it becomes immunocompromised and patient becomes more infection prone. Skin and soft tissue infections like methicillin, gas gangrene and food poisoning. Gas gangrene caused by the Clostridium perfringens and some other species of Clostridium, and it's a membrane disrupting, right? Again, the one exotoxin like cytotoxin cause massive and red blood cells to destructions, and hemolysis takes place. Another exotoxin, enterotoxin, is related to food poisonings and cause diarrhea. So these are the some symptoms and some mechanism of these toxins which are produced by these bacteria, specific bacteria. The antibiotic associated diarrhea is Clostridium difficile, the membrane disruption, and then exotoxin caused by the secretion of the fluids and electrolyte that results in the diarrhea again, and cytotoxic disrupts host cytoskeleton or host structure, and that by damaging the host structures, they can lose everything. The food poisoning is caused by S. aureus, and it's a super antigen, as we discussed just now, and enterotoxin causes the secretion of a fluid and electrolytes that results in the diarrhea. Similarly, toxic shock syndrome is we call TSS, caused by S. aureus, and super antigen toxic cause secretions of fluid and electrolytes from capillaries that disease blood, right? Decreases the blood volumes and lower the blood pressure. So these toxins may cause diseases and discomfort to the hosts by doing different mechanism and different manner, and be successful in establishment of the disease. The features like source gram positive negative and gram negative only. So exotoxins, they are protein in nature, endotoxins are lipid in nature. Exotoxin release secreted metabolic products, and they release on the bacterial lysis. Specificity is very highly specific. Exotoxins and endotoxins are general symptoms. Toxicity, for example, cholera toxin, botulinum toxin, tetanus toxin, diphtheria toxin, they all are very specific and called by specific bacteria, so we call them highly specific exotoxins. Immunity is very strong in anti-toxins, but whereas in endotoxin there's no immunity developed and weak infective antibodies. Heat stability, heat labile exotoxins, but heat stable the endotoxin. Now vaccines which are used is toxoids, and in case of endotoxin there's no vaccines can be synthesized. Now exotoxins and endotoxin again compared here, bacterial sources, and this is the table showing here. So you can go to the table, and some of the things which we have not discussed in earlier slides you can look into here, but like lethal dose, in case of exotoxin lethal dose is very small, but in case of endotoxin the lethal dose is considerably large. So it means large amount of bacteria or toxins are required to cause damage, whereas in case of exotoxin is in a very, very small and micro and less than microgram of drug, these toxins are required to damage the human system or host cells. So this is some references of the book where you can go through it and read about these topics of the virulence and this introduction. Now I can summarize the whole lectures in a few minutes, that the virulence and pathogenicity, as we discussed, that is primary factors for making the bacteria or viruses to be more pathogenic. If you take example of influenza virus, or if you take an example of the influenza virus A or any strain, then it causes damage to the host tissue through HA and NA. So HA and NA are the virulence factors. Now if you reduce the HA and NA through some drug to inhibit this hemagglutinin or to inhibit the neuraminidase activity, then probably you would be successfully block the infection process of this by these viruses. So what is most important here is to understand the different virulence factor. There is an n number of virulence factors present in the bacteria. If you block one virulence factor, the other virulence factor may also work to as a defense system for the bacteria or for the pathogen to be defended by the host system, and they evade the host system. So as we know, the reason of the factors responsible for the infection control is this, that the host get penetrated by the pathogen, and pathogen get entered, proliferate, get mutations, and also damage to the host cells and escaped out, evade out from the host immune system. So this is the whole bottom line, a whole summary of the lecture, that if this is the mechanism, and if you somehow intervene these, by modulating the immune system, you reduce the virulence factors of the pathogens, and you can successfully escape out from the effect of the pathogens to the host system. Okay, so this is all about the infection control, reason of infections, and the pathogenicity, and then the virulence factors and how we can control the virulence factors to reduce the pathogenicity. Okay, in the next topic we talk about the more detail about some of the pathogenicity, reason of pathogenicity, islands, and so on and so forth. Thank you very much.